We are doing NCERT exercise question number 10.21 to 10.25 for the chapter biomolecule. The very first question, what are nucleic acids? I mentioned there two important functions. Now, all living organisms are made up of smallest unit we call as cells. So, in all living cell, in the center we have nucleus. And inside the nucleus, we have nucleoprotein or chromosomes, which are made up of proteins and the nucleic acid. Okay, so basically these nucleic acids are long chain polymers of nucleotide. What are nucleotide? Nucleotide consists of pentose sugar, base and phosphate moiety. So all these things, three things, when they are combined together, they form a nucleotide as a monomer, and when this nucleotide form a polymer, it is called as polynucleotide. Okay, so a nucleic acid is basically a polynucleotide. Nucleic acids are of two types, DNA and RNA. DNA, that is deoxyribonucleic acid, and RNA is the ribonucleic acid. Now, two main function of nucleic acids. Nucleic acid, the very first function, the inherent characters from one generation to another generations are transferred via DNA. So, DNA is responsible for the transmission of the inherent characters or for the heredity. And DNA undergo replication, that means it produces carbon copy of itself at the time of cell division. Therefore, two identical DNA strands are transferred from the mother cell to the two daughter cell. Okay, now DNA consists of a coded message for protein synthesis, but DNA is not involved in protein synthesis. The protein synthesis is actually done via second type of nucleic acid, that is RNA. And there are three types of RNA which work together to do the job of protein synthesis. These are messenger RNA, ribosomal RNA and the transfer RNA in the cell. So this is all about nucleic acid and the function. Second question, what is the difference between a nucleoside and nucleotide? We have discussed that nucleic acid is a polynucleotide. Okay. So, nucleoside, a unit formed by attachment of a base to 1' position of a sugar. Sugar may be ribose or deoxyribose by beta linkage. So, a nucleoside is basically consisting of a sugar molecule and a nitrogenous base, and they are joined to the first 1' position of the sugar. Now, sugar molecule consists of carbon atom as well as the nitrogenous bases are also made up of ring of carbon atom. So, just to differentiate between carbon of a sugar and a base, we number the carbon of a base as 1, 2, 3 and so on, while for sugar we write 1 dash, 2 dash, 3 dash and so on. So, we call it as 1', 3' and so on. Okay. So, nucleoside is made up of only two units, sugar and the base joined by a beta linkage. Now, when this nucleoside is linked to phosphoric acid group, then it is called as nucleotide. Okay, so nucleotide is nothing but nucleoside along with a phosphoric acid group, and this group is attached by the process of esterification of OH group at the 5' position of the sugar. So, for sugar at 1' position we are having your base, and at 5' we are having this phosphoric acid group. So, this is a nucleotide. It consists of a sugar and base just like nucleoside, but it has also a phosphoric acid group or phosphate group. Here we have compared the actual structure of the two. This is nucleoside, and at 1' position of the sugar a base is attached. And in case of nucleotide, at 5' position via esterification, your phosphoric acid group is attached. So, nucleoside have two, is formed by the combination of two type of molecule, while your nucleotide is having three type of entities. Next question. The two strands in DNA are not identical but are complementary. Explain. Okay, we know that DNA has got a double-stranded helical structure, and in this structure, two polynucleotide chains are wound around each other, and they are held together by hydrogen bonds between the pair of bases in the DNA. Okay, now these two strands, we say that the two strands are complementary to each other, because the hydrogen bonds which are formed between specific pair of bases. Okay, so it is not like any base in one chain can combine any other base in other chain. A specific base pairing is there, and why we have a specific type of base pairing? Because of the different sizes and geometries of the bases. Okay, so what we have, what is the condition? Condition is that your adenine, just a moment, your adenine form two hydrogen bond with thymine. Okay, adenine form two hydrogen bond with thymine, while your cytosine form three hydrogen bonds with guanine. Your DNA has got four type of nitrogenous bases: adenine, thymine, cytosine and guanine. Okay, so we are having a specific type of pairing. Your adenine form two hydrogen bond with thymine, and your guanine form three hydrogen bond with cytosine. So this arrangement we have shown here. And due to this base pairing principle, the sequence of bases in one strand automatically fixes the sequence of bases in the other strand. So it's very obvious when restricted type of pairing is available is possible. So we have a set type of arrangement, and therefore the sequence in one strand is always detectable with the sequence of the complementary strand, or the two strands are complementary to each other. Now here we have given an idea about the bases. Nitrogenous bases are of two types. They are purines and pyrimidines. Purines are those nitrogenous bases which have two rings in their structure, or two types of carbon rings, while pyrimidines, they have only one ring. Your purine are two examples are adenine and guanine, while the pyrimidines are cytosine and thymine. Okay, so one purine can form bond with one pyrimidine. So therefore adenine form bond with thymine, and in this case we have two hydrogen bond, while guanine form a bond with cytosine always. So we have three hydrogen bond between guanine and the cytosine. And here we have shown a double strand helix structure of the DNA. This is one polynucleotide chain or backbone, and this is the second polynucleotide chain or the backbone, and these are the nitrogenous bases. So have a look here. The cytosine is bonded to guanine by three hydrogen bonds, while adenine is bonded to thymine via two hydrogen bonds. So therefore the sequence of the bases in one polynucleotide chain is complementary to the one in the other strand which is wound with this. Therefore we say that the two strands in DNA are not identical but are complementary. Okay, next question. Write important structure and the functional difference between DNA and RNA. We have studied that the nucleic acids are of two types, DNA and RNA, that is deoxyribonucleic acid and the ribonucleic acid. So why there is a difference in the name? It is because of the different type of sugar in the two cases. If we look for the structural differences between the two, in case of DNA, we are having your beta D2 deoxyribose sugar, while in case of RNA, we are having beta D ribose sugar. Okay, so have a look here. We have numbered the carbon of the sugar as 1 dash, 2 dash and so on. We have discussed already. So the second carbon, this second carbon is having both the carbon is having what? There is no OH group on second carbon, otherwise every carbon is having a OH group, while in case of ribose sugar we are having OH group on the second carbon. So the difference between the two is that your deoxyribose sugar is not having OH group on second carbon, therefore it is called as deoxyribose sugar because we have removed oxygen. Okay. Now DNA has got a double stranded alpha helix structure. This structure we have already discussed here. This is double stranded. We are having two polypeptide chain wound around each other, while RNA has got a single stranded alpha helix structure. That is only one strand of polynucleotide is there. Now DNA contain two pyrimidines, and these pyrimidines are cytosine and thymine. We have already discussed. But in case of RNA, one pyrimidine is same as DNA, cytosine, but the other one is uracil. Okay. Now we have functional differences also between the two, and the first one is that as we have discussed already, that DNA is capable of producing carbon copy of itself. So DNA has a unique property of replication. Okay, while RNA itself does not replicate. DNA is a chemical basis of heredity. That is your genetic characters or the inherent characters are transferred from one generation to another, and therefore it acts as a reservoir of the genetic information. While RNA does not play any such type of role, RNA is mainly involved in the protein synthesis in the cell. So whenever protein synthesis is required in a cell, or there is need of protein molecule, then the message has been given via DNA. DNA has got the coded message, and then the process of protein synthesis is done with the help of RNA molecules. So this is all about the difference between DNA and RNA. Next question, what are the different type of RNA found in the cell? We have already discussed that there are three types of RNA which are found in the cell depending upon their location structure where they are found. So the RNAs which are found around ribosome, they are called as ribosomal RNA. The RNA which contain messages, they are called as the messenger RNA. And the RNAs which transfer any information or any molecule, they are called as the transfer RNAs. So three type of RNAs molecules are there. This is all for today, and this finishes this chapter biomolecule. Thank you.